So welcome back everybody to Biochemistry 311. Today we are going to pick up from where we left off last Friday, and we're going to talk about the control of gene expression. So just a quick run through of what we talked about last day. We went through the process of transcription for both RNA polymerases found in prokaryotes, as well as contrasting the differences and similarities with the eukaryotic RNA polymerases. So there's a couple of differences. The eukaryotic polymerases have many more subunits that we talked about. There's 3 different kinds, and there's multiple promoter regions that are recognized by these specific polymerases. And then we also talked about termination of transcription in prokaryotes. And we're going to focus on transcription termination in eukaryotic cells today as well. So we'll cover that in addition to control of gene transcription. And once we get through these last remaining slides, we'll start to delve into splicing. And we'll look at how eukaryotic transcripts are processed, in particular the mRNA transcripts, which encode for or translated into specific proteins. All right, so how do we control for gene expression? So in the cells, we have a lot of proteins that are made continuously. And the genes that synthesize these proteins are said to be constitutively on, so always on all the time. They're also identified as housekeeping genes, so proteins that are transcribed and translated continuously. However, there are genes that can be turned off and turned on only under certain conditions. So we talked about an example when we mentioned the heat shock proteins in the prokaryotes. So you can regulate protein amounts not only at the transcript level, but also at the gene level and also at the translational level. But because we're talking about RNA transcription, we're going to focus on gene control of the control of gene expression at the transcriptional level. Okay, so let's move on and just delve into the terminology before we start talking about a specific gene regulatory system. When we have a gene that is negatively regulated, we infer that transcription is prevented by a protein called a repressor. So this protein, in its absence, will allow for the transcription for a particular gene. So this is negative regulated gene expression. When we have transcription occurring in the presence of a protein, it's typically called an activator. And in the absence of this activator, transcription does not occur. So this is often referred to as positive gene regulation. These proteins, the activators and the repressors, as we can see in statement number 3, are allosteric proteins. So do you remember back in Biochem 210 when we talked about allosteric proteins? It means that they can also be regulated at a specific site other than their particular active site. So these repressors and activators can bind ligands at their allosteric sites. Okay, and these ligands can either have a positive or negative effect on either the positive gene regulation or the negative gene regulation. So we're going to look at a couple of examples so that we can explain the terminology a bit better. I'll just advance the slide here. Okay, so when we talk about positive transcriptional regulation, so again, we mentioned that we need an activator, and this will facilitate increasing transcription by the RNA polymerase. So in this diagram, we can see that we have our DNA. We might have, if this is a eukaryotic cell, a Tata box depicted here, and our RNA polymerase coming in to be able to transcribe the particular gene of interest. In the absence of the activator, there is no transcription. However, when this activator binds a particular ligand, so this will bind at its allosteric site on the activator. Okay, this will facilitate increased binding of the RNA polymerase at the TATA box region and increase the efficiency of transcription for that particular gene. Okay, so binding of the activator promotes transcription by RNA polymerase. Okay, and likewise, we can have the activator, in the absence of a ligand, facilitate RNA polymerase transcription. However, when the ligand does appear, so if we add in some ligand, this will negatively influence the activator, cause its dissociation from the promoter region, and cause the RNA polymerase to stop transcribing. Okay, so the activator is essentially needed in both conditions, depending on the function of the ligand. It can either be positive or negative. Positive to allow for transcription, negative to inhibit transcription. Okay, so that's positive gene regulation. For negative transcriptional regulation, it's the same type of phenomenon, but this time we are using a repressor protein. So for this example, we can have the repressor binding to the promoter region, which then this is essentially prevents the RNA polymerase from recognizing that particular promoter region to carry out gene transcription. In the presence of a ligand, so we get ligand binding to the repressor, this causes a significant conformational change in the protein, causing it to dissociate, freeing up, so if this was a eukaryotic cell, the TATA box region, and allow for RNA polymerase to bind and carry out gene transcription. Conversely, the repressor may not be able to bind unless the ligand is present. So in the absence of that particular ligand for the repressor protein, RNA transcription happens. Once that ligand binds to the repressor protein, we get binding to promoter region, the inability of the RNA polymerase to carry out transcription, and hence we stop gene transcription of that particular sequence. So in the absence of a ligand, the repressor does not bind to the DNA. Repressor binding only occurs in the presence of a ligand. Okay, so that's the nomenclature. Okay, so now we're actually going to provide an example of gene regulation, and we're going to actually examine it in one of the best understood gene regulatory systems. And this system actually pertains to lactose metabolism in E. coli, and it's often referred to as the lac operon. So probably you might have heard that terminology in bio during first year or even in high school, I'm pretty sure. So let's just define what an operon is. It really consists of, as depicted on the slide, transcriptional control regions and a set of related structural genes that are basically organized in a contiguous linear fashion along the chromosome. So for the lac operon, if we were to take this terminology, it's best defined as three genes, so a set of genes, that are transcribed from a single promoter region to produce a large pre-mRNA molecule that contains three separate protein coding regions. Okay, so this is just an overall general depiction of what an operon consists of. So let me just write operon. You usually have your structural genes side by side in the chromosome. You're going to have some control sites, so usually the promoter region. And this promoter region is actually controlled by a regulatory gene, so either a repressor or an activator, and it's usually found upstream. So UP, upstream of the operon itself. Okay, so in figure B, we actually have the lac operon chromosomal structure. So we have three genes, the lac Z gene, the lac Y gene, and the lac A gene. And they're arranged in a continuous fashion that are under control of a particular promoter. So all of these genes will be transcribed continuously once the RNA polymerase recognizes that promoter region. There is another operator region, and this operator region is actually for binding of a repressor protein. Okay, so our repressor will bind there, and the repressor is actually defined by the LAC-I gene, which is found upstream of the lac operon, and the LAC-I gene has its own promoter region, so the P stands for promoter. Okay, so the regulatory gene in this operon, the lac operon, is found upstream of the actual lac operon, the three genes, the lac-Z and lac-Y and lac-A. The lac operon is under the control of its own promoter region, and there is a particular operator region that allows for binding of the repressor protein, which is transcribed from the lac-I regulatory gene sequence. Okay, so let's just define what these particular genes are responsible for. So if we look at lac Y, Z, and A, and we're also going to look at lac-I as well in this diagram. The lac-Z gene encodes for the protein beta-galactosidase. Okay, this protein is responsible for, and as we noted before, the operon helps with lactose metabolism in E. coli. This enzyme, remember it's an ASE, which will define enzyme characteristics, breaks down lactose, so the disaccharide, if you remember back to biochemistry 210, into the corresponding monomeric sugars, galactose and glucose. Okay, so lactose is a combination of galactose and glucose. The lac-Y gene is responsible for synthesizing the galactosidase permease protein. And this protein is a transporter, which helps to bring in the lactose into the cell. And then finally, the lac-A gene will be responsible for synthesizing the galactosidase transacetylase protein. And again, it's an enzyme, and it's responsible for acetylating any unhydrolyzed lactose in the cell, and that will lead to its elimination. So you can appreciate how when lactose is present, it only makes sense to synthesize the three transcripts all at once, so that one is responsible for breaking down the lactose, one is responsible for bringing it into the cell, and one is responsible for acetylating it to remove any non-used lactose and target it for elimination. Okay, so we talked about the promoter region over here on the operon, but we also mentioned that there's an operator region. And in this operator region is where we are going to have repressor binding. So the protein that actually binds the operator region is a repressor. And the repressor is synthesized, as I mentioned, from the lac-I gene, which is found upstream of the lac operon under the control of its own promoter. So once we get lactose present, we'll get synthesis of the lac operon. In the absence of lactose, we have synthesis of the lac-I gene, so that we get the I mRNA, which is then translated. So this is a translational process, which we'll talk about in subsequent lectures, to get the repressor protein. One key thing to note is these repressors and activators, or that regulate gene transcription, usually bind as dimers to their particular operator regions. So it's a common theme in gene regulation. So the repressor protein will bind as a dimer and prevent transcription of the lac operon. And in this sense, it is said that the lac operon is negatively regulated, okay, by this repressor protein. All right, so how do we get gene expression in the presence when we have the repressor bound all the time and the gene is negatively regulated? Well, as we mentioned, these repressors and activators similarly will bind a ligand. So in this case, when the repressor is bound and a ligand is present, so this inducer is also your ligand, so just another terminology for it, we get binding to the repressor protein and then a dissociation from the operator region, so it dissociates. And then it doesn't bind with the DNA, and then it allows for the RNA polymerase to come in and transcribe the lac operon. Okay? So once the repressor is not bound, we get synthesis of beta-galactosidase, we get synthesis of the permease protein, and then we also get synthesis of the galactosidase transacetylase. All right, so this, again, still negative regulation by the repressor, but when you bind a ligand, or in this case as the inducer, it will cause a significant conformational change. So significant conformational change. So conformational change in the protein, so in the repressor, dissociation from the promoter region, binding of the RNA polymerase, and then finally transcription of the lac operon. Okay, so we talked about the ligand binding the repressor and then causing its dissociation from the DNA and allowing for the RNA polymerase to carry out transcription of the lac operon. But the question then becomes, well, what is this ligand or inducer as depicted on the previous slide? Well, the ligand is actually allolactose. So it's down here. Okay? And allolactose is generated by not only the enzyme responsible for breaking down lactose into galactose and its glucose monomeric structures, but it also carries out a side reaction. And that side reaction is just really changing the glycosidic link between these two sugars from a 1,4, so 1,4 glycosidic link in lactose, to a 1,6 glycosidic link in allolactose. So allo refers to just another, it's the Greek word for other, so another structure of lactose, basically changing the glycosidic linkage. So it's not a major reaction, just a side reaction that occurs when lactose is present. We get the generation of allolactose. So we get some converted to allolactose, and then this can bind the repressor and allow for gene transcription of the lac operon. Okay, so allolactose will bind tightly to the lac repressor and cause a conformational change that reduces the affinity of the repressor for the operator region, and then hence freeing up the promoter region, allowing for the RNA polymerase to bind tightly and carry out transcription of the lac operon. Okay, so this slide just summarizes what we just went through. The lac operon is a set of negatively regulated genes, so the lac Z, Y, and A. The repressor when it's bound, so the repressor when bound to the operator region will prevent the transcription of the lac operon. There's the ligand, which is allolactose. It's a side reaction carried out by the beta-galactosidase enzyme. When it binds to the repressor, it will cause a conformational change in this repressor, decrease the affinity of the repressor. So decrease the affinity of the repressor for the DNA and allow for RNA transcription to occur. So we basically have just described the first control mechanism through the regulatory response to lactose, which basically is the synthesis of allolactose, the ligand binding to the repressor, region, to the repressor protein, sorry, and then allowing for RNA polymerase, so RNA poly, to carry out transcription. Okay. We're also going to touch on a secondary control mechanism, and that's through the regulatory response to glucose, which we'll see in just a few minutes. All right, so this diagram will actually depict in a cartoon fashion what happens in 3D when we have the lac repressor binding. So as we mentioned, we talked about the operator region that's found near the promoter region, which is found right over here. One thing that the lac operon structural gene figure didn't show is that there's actually a second operator region at the end of the lac operon. Okay, so for each operator region, so there's 2 operator regions. Each operator region will bind a repressor dimer. Okay. These 2 repressor dimers will then come together. So the 2 of them come together and form a tetrameric structure, as depicted in the figure. Okay, so one repressor is found near the promoter region. The other repressor is found at the end of the lac operon. So within this region here, we actually have our lac Z, our lac Y, and our lac A genes that are to be transcribed when the RNA polymerase is able to carry out that function. So binding of the repressor to operator region number 1 as a dimer, plus the binding of the repressor to operator region number 2 as a dimer, then leads to looping of the DNA such that the lac repressor becomes tetrameric in structure. And as you can effectively see, this prevents the RNA polymerase, which is depicted here in blue, from actually transcribing the lac operon, which consists of the lac Z, Y, and A genes. So it blocks transcription very efficiently, this repressor protein, as a tetrameric structure. The good thing about it is that the binding is transient, so it does come off, but then binds very quickly again. However, when it does dissociate, and prior to the binding of another one, a tetrameric structure, within a short amount of time, it allows for the RNA polymerase to carry out transcription. Just go back to my pen. Transcription of this region at least once. So you get transcription of at least once. At least one time. Of that operon, and then hence this actually leads to the basal levels of the proteins that are generated from the lac Z, Y, and A genes in the cell. Okay, that's why we have basal levels of beta-galactosidase, the permease, and the transacetylase, because the tetrameric repressor actually does come off just for a short amount of time, and prior to binding again, the RNA polymerase is able to carry out at least one round of transcription of the operon to ensure that there are basal levels of the 3 proteins within the cell. And you can actually do a pretty neat experiment in the lab, creating your own synthetic operator regions within a strand of DNA, and being able to visualize the repressor protein in action. So in these electron micrograph figures that are depicted on the slide, you actually have created a synthetic lac repressor binding site within the double-stranded DNA. And one of the sites that was actually created is at the 5' end of the DNA, and then introduced a second site down here at about 535 base pairs away. So you can take these synthetic DNA strands, introduce the sequences for the operator region, one at the 5' end of the DNA and another at about 535 base pairs away, and then add in some repressor protein into your experimental assay. And then you can see that looping actually occurs when a tetrameric structure forms of the repressor, allowing for the two sites to actually come together. So here's a great representation. So this is with the repressor protein. And you can actually visualize these using electron microscopy and obtain these types of pretty cool diagrams or figures of the repressor creating these DNA loops as a result of generation of this synthetic DNA. Okay, so we've actually just depicted how the repressor binds in a cartoon diagram, but we're actually going to focus now at the molecular level and see what's going on when we have the repressor binding. So this figure will demonstrate how these interactions occur between the repressor and the double-stranded DNA. So in this figure, we have our DNA represented by this structure here. The region that is presented is the operator region. And again, we have our two repressors binding. So we have one over here and another one over here. And remember, they bind as dimers, so that's a recurring theme. Another recurring theme is that they will bind through an axis of symmetry, as we'll see in a couple of slides. So we have an axis of symmetry for the binding to take place. And the key thing I want you to note is that the interactions between the repressor and the operator DNA region is carried out by this recognition helix. So this alpha helix is also called the recognition helix. And we talked about the recognition helix. Does anybody remember where? When we focused on the RNA polymerase. So the sigma subunit also has a particular recognition helix that makes these transient hydrogen bonds. So we see this green line as a hydrogen bond. Using the functional groups from the amino acid side chains found in the protein, and the Watson-Crick base pairs that are found in the DNA. So once the correct hydrogen bonds are made between these functional groups on the amino acid side chains with the corresponding Watson-Crick base pairs that are contained within the operator region, then you have repressor binding. The helix that's actually makes these types of connections is part of a specific DNA binding motif called the helix turn helix. And we'll look at these motifs in a few minutes. So this is one example of a key structural feature that is required in this DNA binding proteins for recognition along this axis of symmetry. Okay, so on this figure, we have an arginine residue. Again, remember, arginine has some amino groups as depicted here and here. hanging off its side chain, and it allows it to donate hydrogen bonds to the Watson-Crick base pair depicted in this figure. Okay, so we can actually visualize the repressor bound and not bound, and what happens with the helix turn helix motif in this figure. When there is no lactose present for the E. coli, the repressor is bound, and that's depicted as this yellow component in the figure that is associating with the DNA. And when it is bound, we have a clear helix turn helix motif interacting with the operator region in the DNA. When lactose is present, remember we get the synthesis of the ligand allolactose and binding to the repressor, which then leads for its dissociation. So dissociation from the DNA. And if you look at the dotted figure back here, you can actually see that the helix turn helix, so we'll call it a helix turn helix, becomes disorganized. So significant structural changes in the repressor protein, and this is how we get the dissociation from the operator region in the presence of lactose. So the significant conformational changes upon allolactose binding result in repressor dissociation and then hence transcription of the lac operon. Okay, so we can get synthesis of the beta-galactosidase enzyme, and again the chemical reaction that this enzyme carries out is depicted across the top on the slide. It will take the lactose, which is the energy source that's available to E. coli, and break it down through simple hydrolysis using water to its single monomeric structural saccharide units, galactose and glucose. And you can actually visualize the production of beta-galactosidase and the corresponding growth of cells in media once you add lactose and then finally remove it. And that's depicted in this graph across the bottom, where in an experimental condition, E. coli was provided lactose for energy as a food source, and then removed after a certain amount of time. So maybe it was 1 day or 2 days, depending on how long this experiment took. And the measurements of how many cells grew within that time span, so the bacterial protein in milligrams versus the amount of enzyme that was synthesized, revealed that there is a direct linear relationship between the two. So relationship between the two, and when the lactose was present. Okay, so it's a pretty clever experiment that they did in the cell. One thing I do want to introduce here is the terminology of the lac operon. Remember, we're synthesizing a pre-mRNA that has 3 separate coding regions for the lac Z and the lac Y and the lac A. These transcripts are often said to be polycistronic, so consisting of more than one protein coding region. Okay, so that's a terminology that's associated with prokaryotic mRNA transcripts. Okay, so we talked about lactose regulation and how the repressor regulates the lac operon through negative control. There's also positive control of the lac operon. And of course, this positive control is going to be associated with an activator protein. Okay, and we're going to identify this activator protein in just a minute. And this activator protein is dependent upon the catabolite. And the catabolite here is when we're breaking down lactose into galactose and glucose, we're going to regulate it through the catabolite glucose. So when we break down the lactose, we get galactose and glucose. And this glucose, the levels that are present in the cell, can regulate this activator protein, which will have a positive control on the rate of RNA polymerase transcription of the lac operon. And this catabolite activator protein, or CAP as it's often called, will facilitate or increase the efficiency of transcription of the lac operon. Okay, the CAP, catabolite activator protein, and we'll use the acronym CAP from this point forward, is a dimeric structure. Again, here is that common theme that's occurring for DNA binding proteins. It has 22.5 kilodalton peptides associated with it. And a very unique feature in that at the N-terminal region of this dimer, for each of those single monomeric polypeptide sequences, you can bind cyclic AMP, and whereas the C-terminus region will bind the DNA. Okay, so again, this CAP activity will be regulated by glucose, which is the catabolite associated with lactose breakdown. Okay, so how does it provide the positive control of the lac operon? Well, essentially the CAP, when it binds to cyclic AMP, it will assist the DNA. Well, when it binds to the DNA, it will assist the formation of the closed promoter complex so that we can start transcription. So it increases the likelihood that transcription will occur. This catabolite repression by the glucose will ensure that the operons necessary for metabolism of alternative sources, again, you have examples, the lac and the gal operons, are repressed until the catabolite is exhausted, so until glucose is all used up, and then you can activate the catabolite activator protein. Okay, so let's see how this works. When we have lactose present in the E. coli, we'll get breakdown to galactose and glucose. This will lead to an increase in glucose, which concomitantly affects the cyclic AMP levels in the cells by decreasing them. And we won't go into that process because it's an extensive pathway involving phosphoenol pyruvate and adenylate cyclase. But just know as the catabolite levels increase, so glucose concentration, so you can probably write concentration here, when lactose is present, the levels of cyclic AMP decrease. Because CAP needs cyclic AMP to be activated, this leads to inactivation of CAP. Okay? Conversely, if all the glucose has been used up, so you start to get decreases in glucose concentration, we get an inverse increase in cyclic AMP concentrations. Okay? This basically will activate CAP, so it will lead to the binding of each monomeric polypeptide chain to bind cyclic AMP at the N-terminal for each of these sequences, protein chains, so the N-terminal, and then allow for the CAP protein to bind in a region that is upstream. Again, this is upstream of the promoter region. So upstream of promoter region. And as you can see in this consensus sequence, we have some inverted repeats occurring in the consensus sequence along an axis of symmetry. It's got a twofold axis of symmetry. Another common theme for the DNA binding proteins. Okay, binding of CAP in this region increases the efficiency of RNA polymerase transcription. Okay, so now we're actually going to look at some experimental conditions and apply the regulation of both the presence of lactose on gene expression of the lac operon and the presence of or activation of the CAP protein in experimental conditions with E. coli. So hopefully on your slides you've actually have this table, and it's not filled in, so we're going to fill it in together. And we're going to go through 4 different types of experimental conditions and hopefully be able to identify what the rates of transcription are, or whether a transcription is occurring in the presence or absence of either glucose, lactose, the CAP protein, which is also known as CRP in some textbooks, so you can actually change this to CAP if you wish, when cyclic AMP is bound, and the effects of the lac repressor when these metabolites are present. Is it bound or is it not, and whether transcription occurs. Okay, so let's look at experimental condition number 1. We are going to put our E. coli in some media that has lactose but no glucose. Okay, so we don't have the catabolite yet. Think about, is the CAP protein bound to cyclic AMP at this point, and we know it is because no glucose is there, so we have high levels of cyclic AMP. Is our lac repressor bound at this point if lactose is present? In this case it isn't, because we've generated allolactose, so no repressor bound. So then the question becomes, do we get transcription with no repressor bound to the operator region? And of course we do. Okay, so transcription is going to happen. Let's go through condition number 2, where we do have glucose in enough concentration in the media, no lactose. The effects of glucose will definitely decrease the cyclic AMP levels, so our CAP protein is not bound to the upstream region from that promoter region for RNA polymerase. We know that there's no lactose, but we have some glucose. Our lac repressor is bound because we don't have the allolactose or the ligand to cause it to dissociate from the operator region. And since we have the lac repressor bound, obviously there's not going to get any transcription of the lac operon in this condition. So for condition number 3, if we have no glucose present, we have no lactose, obviously our cyclic AMP is going to bind to the CAP protein because we have high levels of it. But because we have no allolactose, the ligand for the repressor is still bound. So irrespective of whether the CAP protein is bound to the DNA, the fact that the repressor is there means that we get no transcription of the lac operon. And finally, in the 4th condition, where we have everything present, so we have glucose present and we have lactose present, this means that the CAP protein is not bound to cyclic AMP and not binding upstream of the RNA polymerase promoter region. All right, and because we have lactose, we have allolactose present, which will bind to our repressor and cause it to dissociate. And again, because our repressor is not bound, we're going to have transcription in condition number 4 of the lac operon. Okay, so then the next question becomes, which condition actually provides a higher rate of transcription? Is it condition number 1 or condition number 4? And think about what will increase the rate of transcription. So which has a positive regulatory control on the rate of transcription? And if you remember back, it's binding of that CAP protein, which increases the efficiency of the RNA polymerase binding or formation of the closed promoter complex. Hence, condition number 1 will have the highest rate of transcription when we're comparing it to condition number 4, where we don't have the cap protein bound. Okay, so think about those conditions. You may be given a question where you're asked to describe the rate of transcription under a certain condition for E. coli, either in the absence or presence of glucose and galactose. And then figure out what is occurring with the cap protein, whether it's bound or not, and also with the lac repressor. Okay, so that finishes off prokaryotic gene regulation. So we're just going to switch gears and talk about eukaryotic gene regulation. And if you remember back to last day's notes, we talked about particular features of the eukaryotic promoter regions, specifically for the RNA polymerase II, where we mentioned that usually we'll have a TATA box upstream of the initiation, ribosomal initiation element that forms the core promoter. And then we also had these enhancer regions that were approximately 1 kilobase pairs away from the core promoter region. And I did demonstrate to you that these enhancer regions can exert their effects on the recruitment of the RNA polymerase through the binding of activator proteins, which are depicted here, which then cause protein-protein interactions with the transcriptional factors, which we see down here depicted in blue. So these are TFs that are responsible for bringing in the RNA polymerase. But what I didn't show last day was that these protein-protein interactions actually occur through another multi-subunit complex called mediator. And there's multiple subunits associated with this. And in this sense, with the looping of the DNA and then the binding of activators, which then interact with the mediator, which then interact with the transcription factors, can actually influence the efficiency of gene transcription for the RNA polymerase in prokaryotic cells. So very complex, very highly regulated, and lots of proteins involved, in addition to looping of the DNA for gene regulation. In addition to enhancers, one thing that I didn't mention, you can also have silencers. So silencer regions will bind repressor proteins, much like we saw for the lac operon, and this can negatively affect the protein-protein interactions with the mediator and essentially shut down gene transcription by the RNA polymerase. Okay? So this mediator complex consists of these co-activator proteins that will facilitate the protein interactions from the enhancer regions down to the RNA polymerase. Okay, so what are the key things that are required? Again, we remember the TATA box region in our transcripts that are associated with mRNA production. Very key. Remember, these TATAs are highly conserved. A single mutation in any one of these ribonucleotides will, in this deoxyribonucleotide sequence will markedly affect the rate of gene transcription. And usually it's upstream, so minus 30 to minus 100 nucleotides before the actual initiation site for transcription to occur. This figure actually depicts the representation of that TBP protein that binds the TATA box region. Remember, TBP is part of a larger multi-subunit complex that's found in transcription factor 2D. Okay, and again, we're seeing the same type of theme where we have this twofold axis of symmetry and our helices coming in and making those specific H bonds to recognize the TATA box regions in the double-stranded DNA helix. Okay, so how does this occur? We've mentioned this already with the lac repressor protein. we'll have key amino acid side chains in our DNA binding protein. So here's an arginine and another glycine residue. We get H bond formation between specific Watson-Crick base pairs. So again, here's a T with an A in the region and another C with a G. And when these correct H bonds are made, then you can get increased transcriptional efficiency by the RNA polymerase. These regions, again, have these inverted repeats, so going this way and going this way. Likewise over here and over here. And they're found along this twofold axis of symmetry. Okay, so a common theme that I see again in eukaryotic DNA binding proteins. Okay, so how do gene regulatory proteins recognize these specific DNA binding sequences? So again, these structural motifs, we've already identified one, the helix turn helix, but there's two other different motifs that are commonly found amongst DNA proteins. At least 80% of these DNA binding proteins have one of three principal motifs based on their structure, and they include the helix turn helix, so the HTH motif, zinc fingers, and we'll look at those in a few minutes, and also leucine zipper basic regions, or B, which stands for the basic B zips, they're often called. Okay, so that's where the B comes from. Okay, so again, another representation of the helix turn helix motif that we see in DNA binding proteins. So here would be our DNA binding region. Here's our helix turn helix. And again, there's a key recognition helix. So recognition alpha helix that makes the correct H bonds within the double-stranded DNA. And again, you can see it more at the molecular level in this side figure. And these interactions, remember, are occurring through the major groove of the DNA, the double-stranded DNA. Okay? So again, we're getting interactions with the Watson-Crick base pairs through the functional groups that are associated from the amino acid side chains that project out of the recognition helix. Now, zinc fingers are a bit different in that you still have alpha helices in these structures, but they're basically tandem sets of small domains, which each of which will bind to a key central zinc ion, which is depicted over here. Okay, through conserved sets of either two sets of Cs, so cysteine residues in the protein, or two cysteines and two histidines residues. Okay, so basically in these tandem sets of small domains, an alpha helix from each of the domain will make specific contacts with the edges of base pairs within the major groove. So if we considered this protein structure over here, we have the alpha helix. So here's our zinc ion. Okay, and this is an alpha helix that's part of this region over here that's making specific contacts with the DNA using the amino acid side chains. So here's another zinc finger up here and another depiction of one down here. Okay, so again, zinc fingers usually are just comprised of tandem sets of small domains in the protein that are conserved, either with the zinc ion coordinating to two cysteines and two histidines, or four cysteines altogether. And it's that a key alpha helix from each of these domains that make specific contacts with the base pairs within the groove of the DNA. And then leucine zippers, or B-zips as they're often called. So B-zips basically look like this structure right here, which is a coiled structure consisting of two alpha helices, very long extended alpha helices, that come together and interact through hydrophobic effects. So hydrophobic interactions in this specific region up here. There is another region below here that actually interacts with the DNA, and this is a very basic region. And it's basic because if we actually look at a comparison of several gene sequences that contain leucine zippers, the DNA binding region actually encodes for lots of lysine and arginine residues. So K and R. So K, remember, is for lysine and R is for arginine. Okay, so the proteins that interact with the DNA, or the DNA binding regions, consist of basic amino acid residues. So this is why they're called B-zips. So these are the basic ones. This region is then connected to the long strand that's depicted up here through a 6 amino acid connector region. And then finally, the long strand that we do see here is called the leucine zipper. And it's so named leucine because every 7th residue is a leucine. So if we look at here, we'll have a leucine, and then up here is another leucine, and so on. And this is why, because leucine is a hydrophobic amino acid, the interactions are hydrophobic for this particular leucine zipper region. So every 7th amino acid is a leucine. Okay, so again, we have another one here and another one here. And when you look at the consensus sequences from these comparisons, again, done through footprinting experiments, you basically have a very basic DNA binding region, a 6 amino acid connector region, and then a very hydrophobic region consisting of leucine amino acids that are held together through hydrophobic interactions. All right, so now let's look at an example of eukaryotic gene regulation, and we're going to focus in on hormonal control, as there's classic examples of how hormones can regulate transcription in us particularly. We'll look at an example using the estrogens. If you remember back from 210, estrogens are hormones that are cholesterol derived. The basic structure of the cholesterol molecule is depicted here. We got 4 hydrophobic rings associated with it. For the estrogens, we typically have a CH3, a carbonyl, and a hydroxyl on that position here. This is actually the estrone that's depicted. But we're going to look at estradiol. And again, part of the estrogen family, we know that it's required for development in the ovarian cycle. Because of these hydrophobic rings being cholesterol derived, it diffuses readily across cellular membranes. And it's also a ligand for DNA binding proteins that are involved in DNA expression. And these DNA binding proteins for the estrogens are the soluble receptor proteins found in the nucleus. Or more specifically, the estrogen receptor. Okay? All right. So let's look at the estrogen receptor before we focusing on the regulation of gene transcription by the estrogens, and more specifically estradiol. This is a 3D structure down here in the corner of the estrogen receptor. It's a very unique looking structure. The protein components from the N-terminal to the C-terminal are depicted here, so that we can actually examine the different regions found within the receptor. We know that the protein consists of a transcriptional activation component, which is right here. And again, that is important for binding activator proteins. So activator proteins, which we'll come back to. There is a DNA binding region, and as you can see within this region, there are 2 zinc fingers that are critical for binding to the double-stranded DNA. And we know they're zinc fingers because they're tandem repeats, small domains, that are coordinated to these zinc ions and these 4 cysteine amino acid residues within the protein sequence. And then finally, there's a region where we can bind the estrogen hormone. It's called the hormone binding region. And again, this molecule will fit into this ligand binding pocket. When it folds into its 3D structure, it becomes a crevice for the ligand to fold into. And then if you focus down here, there's a very unique helix that kind of projects outwards, and this is helix number 12, and we're going to come back to this one as well. So it kind of extends downward. These receptors will actually bind to DNA, and the DNA binding sequences are called estrogen receptor elements, so EREs. So estrogen receptor elements. Elements. And this is where this receptor will bind in the DNA sequence. So they have a specific consensus sequence found in the DNA. And likewise again, as the recurring theme is for DNA binding proteins, the estrogen receptor will bind as a dimer to the EREs. Okay? So we need 2 of them to bind to the ERE, estrogen receptor consensus sequence found in the DNA. Okay, so the receptor binds estrogens, and here is the example with estradiol, and again, that carbonyl from estrone is now converted to a hydroxyl, so we have the hormone estradiol, and estradiol fits very nicely into that ligand binding pocket of the receptor. Okay, and binding of the ligand causes a significant conformational change in the structure of the receptor as we go from without ligand to plus ligand. You can see the difference in the slide. The key major component to this conformational change is that helix 12 that we identified previously, whereas without ligand binding we see it extending beneath the estrogen receptor,